Hey, future biologists! Ever wondered why our cells, the fundamental units of life, eventually start to show their age? The answer, in part, lies within those fascinating organelles we call mitochondria. Join us as we delve into the intricate world of mitochondria and their profound connection to the biological process of aging. Mitochondria are ubiquitous organelles found in eukaryotic cells, often referred to as the powerhouses due to their primary role in cellular energy production. Their unique structure is key to their function. Each mitochondrion is enveloped by a double membrane system. The outer mitochondrial membrane is relatively permeable, allowing passage of small molecules. The inner mitochondrial membrane, however, is highly selective and extensively folded into structures called cristae. These cristae dramatically increase the surface area available for crucial biochemical reactions. The space between the outer and inner membranes is the intermembrane space. The compartment enclosed by the inner membrane is the mitochondrial matrix. This matrix contains a concentrated mixture of enzymes, ribosomes, and the organelle's own genetic material. Within the inner mitochondrial membrane reside the protein complexes of the electron transport chain, ETC, and ATP synthase. This is where the majority of cellular ATP is generated through oxidative phosphorylation. This process couples the oxidation of metabolic substrates with the phosphorylation of ADP to ATP. Mitochondria utilize pyruvate derived from glycolysis and fatty acids as primary fuel sources. These molecules are further oxidized in the mitochondrial matrix via the citric acid cycle, Krebs cycle. This cycle generates electron carriers, NADH and FADH2, which then donate electrons to the ETC. The flow of electrons through the ETC complexes pumps protons from the matrix into the intermembrane space. This creates an electrochemical proton gradient across the inner membrane. The potential energy stored in this gradient is then harnessed by ATP synthase. Protons flow back into the matrix through ATP synthase, driving the rotation of its molecular motor. This rotation catalyzes the synthesis of large quantities of ATP. This highly efficient process is the main source of energy for most cellular activities. Beyond the nucleus, mitochondria are the only organelles in animal cells possessing their own DNA. Mitochondrial DNA, mtDNA, is a small, circular, double-stranded molecule. In humans, it is approximately 16.5 kilobases in length. Unlike nuclear DNA, mtDNA is not associated with histone proteins. This naked state makes it potentially more vulnerable to damage. mtDNA is also inherited maternally in most species, a key concept in tracing evolutionary lineages and studying mitochondrial diseases. While most mitochondrial proteins are encoded by nuclear genes and imported into the organelle, mtDNA codes for a critical subset. These include 13 polypeptides that are essential subunits of the ETC complexes and ATP synthase. It also encodes ribosomal RNAs, rRNAs, and transfer RNAs, tRNAs, necessary for mitochondrial protein synthesis. Damage to mtDNA or mutations within it can directly impair the function of these essential protein components. This can compromise the efficiency of oxidative phosphorylation. Consequently, the cell's ability to produce ATP is diminished. The presence of a separate genome, ribosomes, and the ability to divide by binary fission strongly support the endosymbiotic theory. This widely accepted theory posits that mitochondria originated from free-living alpha proteobacteria. These bacteria were engulfed by an ancestral eukaryotic cell over 1 billion years ago. This symbiotic relationship proved mutually beneficial. The host cell gained a highly efficient energy-producing organelle. The endosymbiont received protection and access to metabolic substrates. Over evolutionary time, most of the bacterial genes were transferred to the host nucleus, but a core set remained as mtDNA. The connection between mitochondrial function and aging has been a central focus of gerontology research. One prominent hypothesis is the mitochondrial theory of aging. This theory proposes that the accumulation of mitochondrial damage, particularly oxidative damage, is a major contributor to age-related functional decline. A key aspect of mitochondrial metabolism is the generation of reactive oxygen species, ROS. These are highly reactive molecules containing oxygen, formed as byproducts of normal cellular respiration. Examples include superoxide radical, O2-, hydrogen peroxide, H2O2, and the highly damaging hydroxyl radical, OH. ROS are primarily generated when electrons leak from the ETC and incompletely reduce molecular oxygen. While some ROS production is normal and serves signaling roles, excessive levels lead to oxidative stress. Oxidative stress occurs when the production of ROS overwhelms the cell's antioxidant defense systems. Mitochondria are both a major source and a primary target of ROS. Their close proximity to the site of ROS generation makes them particularly vulnerable. ROS can damage mitochondrial lipids, leading to lipid peroxidation and membrane dysfunction. They can also oxidize mitochondrial proteins, impairing enzyme activity and structural integrity. Crucially, ROS can inflict significant damage on mitochondrial DNA. As mentioned, mtDNA lacks the protective histone proteins found in nuclear DNA. It also has less robust DNA repair mechanisms compared to nuclear DNA. Furthermore, mtDNA is located in a high ROS environment within the mitochondrial matrix. This combination of factors makes mtDNA highly susceptible to oxidative damage. Studies have shown that the rate of mutation in mtDNA is significantly higher than in nuclear DNA. With age, there is a progressive accumulation of point mutations, deletions, and rearrangements in mtDNA. These mtDNA mutations can lead to the production of defective mitochondrial proteins. This further compromises ETC function and ATP production. The resulting inefficient mitochondria may also generate even more ROS, creating a vicious cycle of damage and dysfunction. The accumulation of dysfunctional mitochondria with age contributes to reduced cellular energy levels. This can impair the function of energy-demanding tissues, like muscle, brain, and heart. This decline in cellular bioenergetics is hypothesized to underlie many age-related physiological changes and diseases. Experimental evidence supports this theory. Studies in various model organisms, including Drosophila melanogaster, fruit flies, have demonstrated a link between oxidative stress, mitochondrial dysfunction, and reduced lifespan. Exposing flies to hyperoxic environments increases ROS production, damages mitochondria, and shortens their lifespan. Furthermore, genetic manipulations increase mitochondrial ROS production or impair antioxidant defenses, often accelerate aging phenotypes. Conversely, interventions that enhance mitochondrial function or reduce oxidative stress can sometimes extend lifespan or ameliorate age-related decline. Recent research also highlights the role of signaling molecules in modulating mitochondrial function with age. For instance, nitric oxide, NO, has been shown to influence mitochondrial biogenesis and respiration. Understanding these signaling pathways offers potential targets for therapeutic intervention. Beyond DNA and proteins, specific lipid and cofactor molecules within mitochondria are also critical for function and susceptible to age-related changes. Cardiolipin is a unique phospholipid found almost exclusively in the inner mitochondrial membrane. It plays a vital role in maintaining membrane structure and fluidity and is essential for the optimal function of ETC complexes and ATP synthase. Cardiolipin is particularly vulnerable to lipid peroxidation by ROS. Oxidized cardiolipin can disrupt the organization of the inner membrane and impair the activity of associated protein complexes. Age-related decline in cardiolipin levels and increased oxidation of cardiolipin are observed in various tissues. This contributes to mitochondrial dysfunction in aging. Carnitine, an amino acid derivative, is crucial for transporting long-chain fatty acids into the mitochondrial matrix. Here they undergo beta oxidation to produce acetyl-CoA, which enters the citric acid cycle. Carnitine deficiency impairs fatty acid metabolism, reducing a key energy source and potentially leading to the accumulation of toxic lipid intermediates. All right. Mitochondrial carnitine levels may decrease with age in some tissues. This can compromise the cell's ability to utilize fats for energy. Carnitine also interacts with cardiolipin, potentially offering some protective effects on the inner membrane. Certain drugs can interfere with carnitine metabolism, highlighting its importance. Coenzyme Q10, CoQ10, also known as ubiquinone, is a lipid-soluble benzoquinone. It functions as an essential electron carrier in the ETC, transferring electrons from complexes 1 and 2 to complex 3. CoQ10 also acts as a potent antioxidant within mitochondrial membranes.